So I also want to thank the organizers for this opportunity. I'm going to bring forward some work that is quite relevant to the previous speaker. It's a summary of the program that we've had with the Department of Energy Atomic, Molecular, and Optical Physics program. You might wonder, what does atomic physics have to do with carcinogenesis? Well, it turns out that there's a body of information that has come from that community which is quite relevant to understanding the initial stages of DNA damage. And those stages of DNA damage may be able to be harnessed so that you can develop radiation treatment therapies. So what I'm going to concentrate on is understanding low energy electron interactions with complex biomolecules and its relevance to carcinogenesis, and in the end, its relevance to radiation therapy. The work was done by a number of really good and talented people. Aaron McKee, a former graduate student, and two postdocs, Michael Scheibel and Shramana Kindu. And essentially, most of the work that I'm going to discuss is done at Argonne National Laboratory in collaboration with Richard Rosenberg. This is an important slide here where it demonstrates a number of things that can happen when DNA interacts with radiation. I will focus mostly on the role that X-rays play in damaging DNA, but that's a little bit misleading. I won't talk about what the X-rays are doing specifically. I'm going to concentrate heavily upon what the secondary electrons do that are produced by the interaction and the ionization events that the X-rays do. So the X-rays are a source of slow electrons. And so the whole story is, what does the slow electron do? And in particular, what does the slowest electron do? And most chemists will dismiss this because the energies of these electrons that we're going to be caring about is less than 3 eV. And people will say, gee, that's below the bond energy, so that shouldn't matter. It turns out that they're really, really effective in damaging molecules because they have to go through these things called resonances to finally stop and trap, and it's the resonance scattering problem that leads to damage. Now, let's see if, is there a, yeah, there's a little tiny red thing here. Okay, so, the X-rays come in, they can make a number of products. I'm mostly concerned about what goes on right here. The X-ray can come in, and there's this thing, cisplatinum here. This is a known molecule that can be intercalated into DNA and used to do radiotherapy. And what it does is it leads to sort of local proximity damage in a double strand break. This double strand break is particularly potent. So we've spent a fair amount of time trying to understand what leads to double strand breaks, and in particular double strand breaks that are extremely close to one another, because it is the misrepair of these double strand breaks that lead to cancer. If it isn't repaired and it goes towards apoptosis, that's good. The cell's dead. The problem is it's very often misrepaired. You do this side correctly, that side wrong, and then you have a replication error that then translates into a mutation, which then translates into cancer. And so we really are concerned about what leads to these localized double strand breaks. And we've done a number of experiments where we've looked at long DNA chains and we've been able to unravel the resonance here. But we're going to back up a little bit and see, what's really involved in the scattering with the bases. Just a really simple part of the DNA. And then we're going to pay close attention to the fact that if I have an X-ray come in, I'm going to generate secondary electrons that are going to be proximal and they can lead to damage. Over here, we have to pay attention to this excitation here. So this is an X-ray excitation. It's a core hole excitation. I'm going to use a technique known as X-ray photoelectron spectroscopy, where I remove the electron and I measure the kinetic energy of that ejected electron, and that's used as a very, very local probe of what the structure, the identity of the atom is and the structure around that. So it's a very chemically specific probe of the local structure. What also is important is the local structure is involved in causing different decay channels. Now, anybody who does X-ray spectroscopy will know that one of the primary decay channels is what's called resonant Auger decay. So I have this low energy, the core hole produced. This core hole can be filled in by an electron higher up, and that ejects a lower energy Auger electron. Now, most people stop there. They said, okay, well, I have an ionization event, there's one electron, I have this Auger decay, there's another electron. But there's a very, very important proximal effect that happens that has not really been paid attention to as much as it should, and that's called Coulomb decay. Now I have a sort of shallow core hole, and there's a lot of energy still here, so when this thing decays, what happens is there's this Coulomb interaction, and it could cause the neighbor to ionize. This is a lot of physics. What we really care about is there's a lot of slow electrons that could be made proximal to a target, and those slow electrons damage the material. That's the point. So this is an entry channel to make lots of slow electrons that people don't particularly pay attention to. And I want to show you some work that has been in the literature, so this isn't fantasy. You come in and you can measure in coincidence the ionization channel. If you come in and this is the 2A1 hole of water. It's the level on the oxygen primarily. You can look at that ejected electron in coincidence with a very slow one that's ejected due to this shallow core hole. This slow one is being produced by this Coulomb decay channel. And you can see this in the coincidence map. And what it says is I've got lots of electrons below 5 electron volts, which most people would say don't matter. That's fine. This is the end state of ionization. You make slow electrons, they trap. That's true. But those slow electrons trapping, the process they go through to trap, is going to break the DNA with very high cross sections. Okay? So we had a review article that we wrote, which it's actually, I think, a pretty good article. It brings out a lot of important physics of what goes on. There is a heavy weighting on the inelastic scattering channel. So we come in with these slow electrons. They can attach to the base. You have a resonance here. And then it moves through the DNA, and they end up breaking the sugar phosphate bond. So I capture an electron on the base. The base itself can break. I'll show you this. The sugar itself can break. I'll show you this. And you make a sugar phosphate breakage, which is the DNA strand break. And so you end up down here breaking things, but you start out with an excitation on the base. Okay, this particular review article was very heavy on the base, but I want to show you that that's not quite right. It's a little misleading because you can have direct scattering with the sugar, and you can have direct scattering with the phosphate, and you can have very high cross sections, and they're going to be specific with respect to where you have the damage. They're damaged in all sites, and so this is a very effective way of ripping apart DNA, which is useful if you want to use it as a radiotherapy agent to kill the cell. Okay, I'm going to back up a little bit, and I'm not going to spend a lot of time on this because there's a lot of details. We've written this up. It's in a couple of papers right now, but we used X-ray photoelectron spectroscopy to sort of unravel where this damage happens. And we go to the synchrotron facility at Argonne National Laboratory. It's a beautiful synchrotron. It's very high flux tunable X-rays. We now are doing this work at Berkeley using time resolved X-rays, so that we can measure in real time the lifetime of the holes, where those excitations are on the biomolecules, and try to understand the physics of energy transport in the attosecond time regime, which is pretty fast. It's on the it's on the time scale of motion of the electrons. Now, what we want to do is a trick. It's a simple thing. We use the X-ray to irradiate a film of DNA or a film of nucleotides, and I've drawn this as sort of DAMP. And we wanted to compare DAMP with RAMP. And this is, chemically a trivial difference. There's just a difference of the oxygen on the sugar. And we were wondering if this simple change on the sugar affected the damage probabilities of these. And so we hit it with an X-ray, and then we probed the X-ray photoelectron spectra as a function of fluence and as a function of energy. And so we can, look very specifically where the damage is. So we use the X-rays to generate the secondary electrons and probe the damage simultaneously. So we've looked at the oxygen 1s level, the carbon 1s level, the nitrogen 1s level, and the phosphorus 2p level. And you can see that there is a well-defined change. So the black part of this curve is before radiation, very first run. And then you irradiate it for a while, and then there's a post-irradiation plot here, which is in the red. And then this can be deconvoluted into a number of different components. You can see O1, O2, O3. There's a well-defined process for doing this deconvolution. And this O1, O2, and O3, they have chemical relevance, right? It's the same thing with the carbon. You can see changes in this, and this is deconvoluted into 4 different types of carbon. You look at the nitrogen, there's 2 different nitrogens, and then the phosphorus, there's 2 phosphorus. This is the spin 1/2 and spin 3/2 of the phosphorus. The phosphorus doesn't tell us much. We know it's breaking, but the phosphorus itself doesn't tell us much. What's giving us the most information is the oxygen 1s level and the carbon 1s level. So, the chair of chemistry is here, so I had to put a molecule on the slide here. Normally you don't do that. And so this is the chemical structure of the RAMP. And these O1, 2, and 3, and C1, 2, 3, 4, these deconvoluted assignments map onto this particular molecule. So there's specific sites within the molecule that we can watch the damage of. And so O1 is particularly important. That's on the phosphate group here. The O2 is particularly important because that's on the sugar. And you can see that the different carbons, there's the carbon in this glycosidic bond here. There's carbons in the sugar. There's carbons in the base. And we can look at the different nitrogens, which are this group here versus those within the ring. And so we can unravel all of this and measure the cross-sections of damage in a site-specific manner, which is pretty useful. So we've done that. And the way you do this is, and it's kind of a boring experiment, you sit there and you wait, and you do the fluence dependence and time dependence of this. This is a very difficult talk to give to the AMO physics program. The review is because almost everybody else in the program is very active in attosecond work. And so I had the attosecond people talking right before me. And I really changed this because this was time. And I realized that the time was about 4 billion attoseconds. And I said, well, I better put it in fluence. It's a little bit better. But anyway, you look at the time, the time dependence or the fluence dependence, and you fit these decay curves. And you saw them for oxygen. You see something grow at a rate and something decline at a rate, and you can get a rate of damage and a cross-section for damage. And we've done that, and you can see, there's a difference. Here's the initial, and here's the final. And so you look at the areas underneath these curves or these components, and you can extract out damage probabilities and cross-sections. Okay, so we kind of understand this pretty well, but the problem is I used an X-ray to make the electrons, the secondary electrons, and the energy distribution of those secondary electrons is not so well defined. So we're going to improve the measurement so that we can actually do an energy resolve experiment. And as I mentioned to you, now we're going to move to do a time resolve experiment, and then we'll have all of this mapped out. One thing that it told us, which we didn't really know ahead of time, is that the DAMP and the RAMP, they behave ostensibly the same, so there's no real difference in their radiation stability. And then we can tell what particular sites are being damaged. And you can see that the sugar actually has a pretty big damage cross-section. All right, so the cartoon here is we basically have an electron that comes in. It can scatter You could have it come in and excite the base. It can move through the sugar and end up on the phosphate. It actually can directly scatter with the sugar, and it can directly scatter with the base. And all of these, parts of the nucleotide, they support resonances on their own. Okay, so there's all kinds of resonances that are there, and we can unravel them. But I'll show you something that surprised us, and what it tells us is that they're window states. The resonance that you enter in the initial scattering channel are simply open window states. The final states that lead to damage are the shape resonance at less than 3 eV. So the very, very slow electrons, when you get it to that manifold, it's the very last step in stopping that leads to damage. So a few eV electrons in the wrong place really efficiently break DNA with a very high cross-section. So from a radiotherapy standpoint, what you really want to do is maximize the number density of electrons at very low energies so that you can specifically take out a particular DNA strand. Okay, so we were looking at this and we said, okay, well, we don't know the energies, exactly what the energies are. And we were biased when we did this experiment because we know Feshbach resonances at a higher energy have big cross sections. And so we said, oh, we'll look very specifically at what the Feshbach resonances are. These are words. A Feshbach resonance is an electronically excited collision target. So you have 5 eV or more of energy in the collision. But that electronically excited target captures an electron. So it's a 1-hole 2-electron configuration. You have an extra electron on an electronically excited target. And then that'll decay. And so you have the electron that sort of ends up leaving with low energy, but then you've imparted 5 or 6 eV in the electronic excitation. We thought these were going to be the dominant channels. They are the dominant channels for double strand breaks that are proximal. It's not the point of this talk. What happens, the dominant channel for single strand breaks is the slow electron that leaves when that thing decays. Because then that has to go through what's called a shape resonance that's caught in the centrifugal barrier at low energy. That's long enough for the anion to get on a repulsive potential. We only know this after this experiment. We thought the Feshbach resonances were the key to this, and it turns out that's not true. How do we know this? Because what we did was we took a very similar experiment, but we brought it here. We built it here. We didn't go to the synchrotron. We actually took an X-ray system that our engineering colleagues threw in the dumpster. We fixed it, and then we put on our own electron beam, and we said, okay, we're going to probe this. We're going to irradiate this with a slow electron, which is tunable. So we have a monochromatic electron beam that's hitting the surface. Then we're going to probe the damage with a low flux X-ray source. Do the same experiment we did before, except for the electrons that are doing the damage, we supply them directly and we know their energies, right? Which is useful. So we can do this energy resolve measurement. And it's the same thing. We can see damage with DNA, on the oxygen line, the nitrogen line, and the carbon line, and we do the deconvolution. And we can do this just the way we did it before as a function of flux, but we can do it as also a function of energy. And so we did that, and we had a lot of data. And these, I'm not a real fan of supplemental material, but we had to do that for this paper. And so we showed, that, we do this with lots of different energies. This is just a subset of the energies. And you can see here's the initial, here's the final. And it's interesting. If you look at this, there's these final states that are very similar. If I got 3 eV, which is below any of the Feshbach resonances, it's only shape resonances, then 5 eV, 10 eV, these are ostensibly the same. Similarly on the nitrogen, right? So 3 is behaving like 5, which is behaving like 10, which is weird. If you're a chemist, you would say this shouldn't be the same, right? These are very different energies. But it turns out that they are the same because the 10 EV excitation cascades down to a 3 EV excitation. So that's the bottom line. You can analyze all of these, decay curves, and as a function of the site and as a function of energy. And what we find is that the decay curves are rather similar. The decay curves give you a decay constant which is correlated with the growth. The growth is a COP bond, so that's the primary damage channel here, is the breakage of the COP bond. You get a lot of base damage, you get a lot of sugar damage. So all of the sites that you can break, you will. And it'll happen even at 3 EV, right? And it turns out that the cross sections for this aren't that different either. They do go up and down a little bit, and they go up when you have a Feshbach resonance, but the damage end state is not different. It's just the cross section. And the reason for that is the number of slow electrons you make is a little bit higher. And so in the end, the slow electrons do everything, and they're really effective in damage. So it's like, what does that have to do with radiation treatment? Well, I mentioned this, and we don't need to go through this. All we need to do is believe me that everything cascades down to the slow resonances, less than 3 eV. And so if I really want to affect my damage, I want to be able to get into the cell, and I want to have a lot of 3 eV or lower energy electrons that are at the DNA to break it. And I gave this talk in Sicily, and I had an interesting question, which I'll get to after I go through this particular slide here. Why does this matter here? You have multiple scattering going on here, all of these multiple ionization events. They're all kicking out slow electrons that will continue to scatter. I have this attachment, autoionization, and an attachment. It's this attachment step that matters a lot, right? And the autoionization step, I think, is quite important. It makes this red circle. It's the red circles that have the slow, less than 3 eV electrons. The red circle is also an excited state that I mentioned from a Feshbach resonance. And you can cause the DNA to be damaged rather efficiently. Now, this has been modeled. You can use an excitation that moves through the base into the sugar. What I want to do is show you some stuff that has been done that makes this clear that this is relevant to the cell death. You can put in double strand break DNA. You can look at the survival probability of different cells, and you can show that the DNA double strand breaks and the cluster damage doesn't really deal with explaining the effective death of cells. And it turns out that in this paper here, in JPhys Chem, there's a strong argument that says that these crosslinks, which are more subtle chemical changes, are pretty effective in causing the cell death. And that's related to what's going on at the histones, and then what may be also going on with different proteins. So when I was in Sicily, there was an interesting conference, and there were some clinicians in the audience. And they asked me, they said, well, you know, it was similar. It's like, well, you know, if you want to treat DNA or you want to get this stuff in the cell, it's not easy. We had a really good talk about how it's not easy to get things into the cell. He said, why do you want to put this into the cell? Why don't you just attach or attack the cell membrane? And I said, well, that's pretty interesting because the inelastic scattering of the electrons with the proteins at the cell membrane will be very effective in damaging the proteins. And I said, well, this is perfect. This is in Sicily. I said, this is the Sicilian way of treating cells. You break the legs, okay? You go and you simply break the legs so that they don't move around and you can't get tumors to grow. So we've switched the program a little bit to look at, you know, how this works and how you might be able to damage the proteins, right? And so I don't want to spend too much time on this other than that. You say something really similar. You get a pretty effective cross-section for the damage of simple amino acids and end sites on proteins. We played a game where we were looking at the spin of the electron. And it turns out that not only does the electron presence matter, but the spin matters, which is what you would expect from the physics of the electron scattering interaction. And so controlling the spin would also be good, right? So this is sort of a direction we're going in, is to control the electron energy and spin at a particular point to do targeted therapy. Now, We may not, we will probably want to do this with nanoparticles that enter or adhere to the cells. We're particularly interested in the second part of this, which is adhering to the cells. You know, getting specific antigens on it where they can adhere to the cells, but we don't have to go through a lot of DNA damage. We may be able to simply do some mitigation of growth by taking care of the proteins. Now, this is the most important slide. Okay, I'm very heavily involved in human exploration initiatives with NASA. And when you have human exploration beyond Earth's orbit, you necessarily must pay close attention to radiation effects. There's been a very heavy emphasis on radiation-induced carcinogenesis from high LET radiation from the solar flare events and stuff. But I want to point out something that some people here might be interested in. So we are working already on a major human space exploration initiative within the state of Georgia, which is well beyond Georgia Tech. Okay, it involves Emory University Medical School, and it will involve the University of Georgia. And there are two things that we're immediately working on, and that is, I'm working with folks in the radiation oncology division at Emory, getting together a call and a response to this thing called TRISH. TRISH is its Translational Research Institute for Space Health. Georgia Tech wrote a proposal to get this. It didn't come here. It went to Baylor College of Medicine, Caltech, and MIT. It's about a $150 million program for translational research for space exploration. They have a call out right now that we're looking at to identify countermeasures against ionizing radiation. There's carcinogenesis that's in this, but it's actually really focused towards neuro. It's neuro here and neuro here, and this is coming out right now. It's a 2020 NSCORE call for radiation effects on central nervous system. Beyond the damage to the neurons, they want to look at behavioral effects, impact on cognition, and impact on sensor motor responses. This is going to be a big program, and so we're trying to mobilize now ahead of time to do this, right? Getting things across the blood-brain barrier and seeing what goes across that blood-brain barrier due to radiation is wide open. Nobody knows how to do this, at least what I can see in the literature. And so this is something that there might be some interest in program growth and collaborations, and that I'll stop. Thank you. Thank you. And if you have any questions, I will be happy to answer them, and I will be around to talk to anybody who cares about moving forward in looking at programs that are related to human space exploration. Okay. Any questions? How many of you believe that low energy electrons matter? Okay.